if everybody can reconvene and we'll be able to digest more data while we digest lunch. And our next speaker will be Dr. Ed Levin, who I already introduced once, but and who really needs no introduction. He's been a very prolific colleague and is just always amazes me with the breadth and depth of the research that he does in terms of possible new medication treatments, and also looking not just at one system like dopamine, but looking at how multiple systems interact in the brain. And that is what he's going to present, is interacting neurobehavioral systems in tobacco addiction and its treatment.
Okay, well, thanks, Jed. Thanks for inviting me. So the postprandial talk is always the most daunting. I'll rise to the occasion, I'm sure. So I'll be talking about it's complicated. So it's a simple question: why do people smoke? Murray Jarvik, who Jed and I both had the acquaintance of and worked with, raised that question, and that's a good question, because people take in this burning tobacco, which contains nicotine, and I don't know, depending on your limited detection, 6 or 8 or more 1,000 other compounds, and it's highly addictive. Anyone who's engaged in tobacco use will tell you it's no great thrill. So the idea that drugs of abuse are necessarily thrilling is immediately squelched by the case example of tobacco addiction. There are a lot of things that go into it. One is nicotine. One is the act of smoking, the sensory properties of smoking. So I'm going to get into that. First, a caveat. We're not looking at smokers. We're looking at rats with IV jugular catheters, and they're self-administering nicotine that way in a pretty admittedly impoverished environment. They do have sensory cues, a flashing light, but we're trying to expand upon that, and as other people are. But often is the case in science that we have a reductionistic attitude. And I think that's just, it's not unfair to say that a lot of science is reductionistic. We try to find the mechanism of whatever. And I think a lot of people, or too many people, are trapped into this definite article. I tell the students always, indefinite articles are the order of the day, a mechanism, because you never should have the hubris to think you do know it all. You may know a lot, but there's more that you don't know, and there are things that you don't know. But in any case, nicotine. Nicotine was that's the psychoactive ingredient in tobacco. It's certainly a psychoactive ingredient, a very important psychoactive ingredient, not the only psychoactive ingredient. There are certainly other nicotinic congeners that are in adabasine, tabine, nornicotine, and so forth in their MAO inhibitors, and a lot of other bioactive compounds. But let's even just take the case of nicotine. Nicotine does cause dopamine release. That's been shown for many decades now. And some do say, and the current director of National Institute of Drug Abuse says dopamine release in the nucleus accumbens is the final common pathway of addiction. However, I take exception to that, and I'm not the only one, because nicotinic acetylcholine receptors are distributed broadly throughout the nervous system, and actually in non-neural systems. Nicotinic receptors are quite ancient phylogenetically, and they are found on leukocytes, and they're found in muscle tissue. They're found in a lot of different places. So, and they're found in a lot of places in the brain that aren't necessarily directly related to dopamine. And Susan Wannicott and her group among others, have found that nicotine potentiates release of a wide variety of neurotransmitters. And even if it didn't do so directly, the brain is an organ of communication. I think that's a slide a little further along the line. But these neurons are connected to each other. And some say circuits. I mean, that's just a metaphor. I mean, it's kind of not really just like electrical circuits, but they have these systems that are really working in close order with each other. So between our ears is an enormously complex network of interconnected cells. So nicotine affects a wide variety of neurobehavioral systems. So a wide variety of behaviors, not just reinforcement or reward, but also having to do with stress, having to do with sensory function, motor function, having to do with hormonal regulation, and so forth. Unless you want to say, get reduced to the absurd, that the final common pathway is a neuromuscular junction, well, yeah, I could say, yeah, that's the nicotinic system as well. But in terms of things that are upstream of that, it's enormously complex, and I think we should embrace that. And there are a variety of other active compounds in tobacco, as I mentioned, and that the way that tobacco is delivered in terms of this repeated consummatory action, either by smoking a cigarette or a cigar or a cigarillo or whatever, a jewel, it's really a hand-to-mouth sort of thing. But then also in terms of rubbing stuff, having the oral route is particularly important. Nicotine skin patches aren't necessarily abused. Jack Henningfield showed that a long time ago. Nicotine's pretty freely available. It's not shut up as far as I can tell, but it is widely used in terms of cigarette smoking. So the community of neurobehavioral systems underlying addiction. Last year I talked about this, greater metropolitan area of dopamine. So some of these concepts are taking off from that way, but I would be so bold as to say there are no final common pathways of addiction. And I realize that flies in the face of a lot of accepted wisdom of the almighty ventral tegmental to nucleus accumbens dopamine superhighway. But I'd say that that's very important, but it's only a part of what we need to understand addiction. And that there are, as a consequence of no final common pathway, no magic bullets for curing this. And so the fact that you have a variety of reasons for engaging in this addiction, and whether it's the ventral striatum in the accumbens or the dorsal striatum and the habit formation or other areas that we will talk about in just a minute, this is a problem, but it's also an opportunity, as they always say in interpreting Chinese characters. So this opportunity is that there are a number of areas to intervene. And there are a number of neurobehavioral systems. I mean, people may be primarily living in different parts of their brain or living in different parts of their brain at different times of the day. So at certain times you're hungry, at certain times you're stressed, and certain times you're thinking, and sometimes you're doing a lot of these things at the same time. But what's primarily driving your behavior may be one or the other or the third of these systems. So again, as I said, the brain is an organ of communication. So not to belittle anybody who studies the liver, but by and large, you've seen one hepatocyte, you've seen most of them, if not all of them. But the neurons come in a wide variety of different types and connection patterns, and we have to consider that when considering normal function and abnormal function, such as addiction. So that addiction is more than dopamine systems, and tobacco addiction also is more than involves nicotinic systems. As complex as nicotinic systems are important. And we have to acknowledge the brutal truth is that current treatments have very low success rates. If most any other drug treatments were as successful as the current treatments for helping people to quit smoking, they'd be termed miserable failures. The fact is that they do significantly help people more than placebo. Placebo rates are very, very low to begin with. But we just have a very long way to go in terms of having efficacious treatments. And I'm going to present to you a series of pharmacological treatments, but it's not just pharmacological treatments that are the answer by any means. And the integration of those treatments with other treatment modalities, be they behavioral treatments or transcranial magnetic stimulation, who knows what, I think is going to be the order of the day. So different people may smoke for different reasons, and also people may smoke for different reasons at different times. There are rewarding effects. There are demonstrable cognitive improving effects, as we heard earlier today in this session, as people have seen in a variety of studies, ranging from honeybees, as was shown, to zebrafish that we've shown, to rats and mice, to people. And that it may be the fact that nicotine facilitates its own addiction in terms of enhancing the secondary reinforcing effects. Stress alleviation is certainly an important aspect in habit. Appetite is something that's the hypothalamus, I think, is largely overlooked. Some people have looked into it, and we have too. And I think that deserves recognition as be a part of this system involved in addiction and withdrawal relief and others. So these different behavioral systems involve different neural systems, naturally, and then combining different parts of the brain systems may help with the efficacy. So I'm certainly not the first one to say this. Tom Itzel made an eloquent presentation in Scientific American some 8 years ago. And just to understand that you have these dopamine systems that not only go from the ventral tegmental area to the accumbens, but also that same mesolimbicortical pathway goes to the frontal cortex, and there's innervation from the nigra to the dorsal striatum, and there's also dopamine innervation of the hypothalamus. And whenever people talk about stress, they often talk about the HPA axis and the hypothalamic pituitary adrenal axis, and they often forget about the H part. H part is hypothalamus. So again, I'm going to come back to that as one of the underappreciated drivers. But there are cholinergic systems that are equally widely dispersed, and especially important is this cholinergic systems, innervation of the VTA, innervation of the habenula, but there's interstitial cholinergic cells in the striatum, dorsal and ventral. There are certainly important cholinergic innervation of the hippocampus that may be important in terms of the secondary reinforcing effects that are key, or I would say essential, for nicotine self-administration and tobacco addiction. Serotonergic systems, certainly, and then many others. And then that there are these interactions among these systems and brain areas that bring in areas like the amygdala, which is key in terms of emotional responsivity. So, in any case, so some of the drivers of smoking, and this is just a partial list, I'm absolutely sure. Primary reinforcement, yeah, do it again. You know, whether it's rewarding or not, it may be reinforcing. You know, I have this bad habit of cracking my knuckles, right? It's no great thrill to do it. It's like, I got to do it, you know. Sometimes you have things that you feel like you have to do. It's a relief not to have to do them after you've done them, but it's not a great thrill having done them. Secondary reinforcement, essential. Derek Donnie has shown it, Jed has shown it, others have shown it, you know, throughout the field that the throat scratch or the aroma, or not just sensory, but the act of self-administration as part of that kind of secondary reinforcement, the act of going through that habitual self-administration, I think, is reinforcing in itself. And habit, you know, my mother used to say, habits can be your slaves or you can be their slaves. So depending on if it's good habits or bad, these kind of automatic sort of things could certainly be. So in that sense, why do you smoke? You can even take out the why part, because there's no sense to it. It's just an imperative that smoke. You can take it down to one word. Stress, certainly that's widely known to cause relapse and the appetitive nature of smoking and certainly withdrawal effects. So there are certain areas of the brain seem to be important in some of these drivers, like nucleus accumbens with the primary reinforcement. This is Gina Corelli's data looking at dopamine release. This is with cocaine. And actually, she found with the secondary reinforcement, that itself will cause dopamine release after the training. Secondary reinforcement in terms of learning aspects, the limbic system, among others, frontal cortex as well, certainly should be considered as being very important. Dorsostriatum in terms of habit, stress, the amygdala, hypothalamus, and appetitive in terms of the hypothalamus. Dick Kesey, who was a professor when I was a graduate student at Wisconsin, did some key work in terms of the ventral medial and the lateral hypothalamus and feeding mechanisms. But in terms of their driving the appetite for drugs, I think that's an important aspect as well. And then withdrawal in multiple areas. So I'm going to go through a series of studies where we've looked at nicotinic, D1, D2, serotonergic, 5-HT2C acting drugs, histamine, the kind of ignored monoamine is important, glutamate, and especially NMDA, this ligand-gated ion channel of the glutamate system, and norepinephrine alpha-2 drugs. Not to say this is exhaustive, but this is what we've done over the last few years. So in terms of acetylcholine, this is more of a validation of the procedure of the rat model than anything else, because we know nicotine skin patches do help people quit smoking. Here we have chronic nicotine administered through osmotic mini pumps, significantly reduce nicotine self-administration in a dose-related manner. So that's a good validator. Varenicline, which is also known to help people quit smoking, does significantly reduce nicotine self-administration in terms of dose-related reduction with varenicline. Dopamine systems, we've done a lot of work with D1 and D2 drugs. Shearing 23390 is really an old line D1 antagonist. All these drugs, whenever I ascribe a certain effect to them, it's with the caveat that no drug is clean. Every drug is started off being clean, but gets dirtier as people investigate it further. And that meaning it was dirty all along. They all have multiple effects, but it does have a predominant effect on D1 receptors, and it's an antagonist. So that significantly reduces nicotine self-administration. And haloperidol, the D2 antagonist, does not really. We've done a whole series of local infusion studies with D1 antagonists and D2 antagonists infused in different brain areas. Here's one in terms of the insular cortex, a significant reduction in nicotine self-administration with D1 but not D2 in the insula, and then also in the nucleus accumbens shell. I'm not showing the null effects of haloperidol, but we've driven it up to the limits of solubility in terms of local infusions, seen nothing in any of these areas. In terms of the parietal association cortex, this may be something new to the addiction systems that people are considering, but you know, this posterior parietal cortex is kind of an interesting area in terms of motivation. There have been some studies of people who are awake undergoing neurosurgery for taking out epileptic foci, and they need to stimulate certain areas to see which areas to take out and which areas are important. And whenever they do the motor cortex, your arm will rise, and a corresponding area in the posterior parietal cortex, you want to raise your arm. You don't just raise your arm, but you want to. So that as a motivator, as a seat of impetus to action, I think is really under-considered as far as a driver of behavior in general and addiction in particular. And you know, a lot of people think the frontal cortex is the front seat driver, but in some sense the posterior parietal cortex is, you know, your back seat driver telling your front seat driver what to do. Anyway, anterior cingulate, it wasn't quite significant. It was a more modest or minor effect, and certainly in our study it didn't reach the level of robust effect that would achieve significance with the study we did. The hypothalamus, this is supramammillary nucleus, had a pretty dramatic effect. And again, in all of these areas, D2 antagonism had no action whatsoever. With serotonin, we worked with lacasterin, which is 5-HT2C agonist. It's marketed currently as a treatment for obesity, and it does help people lose weight. And then we also saw that it significantly reduces nicotine self-administration. We're not the only ones. Other people have found this as well. And people have found that it reduces cocaine self-administration as well, and acutely and chronically. And there's a recent human clinical study that Jed was part of, and they published that significantly reduces nicotine or tobacco smoking as well. So that area, interestingly enough, we tried to find an area of the brain where it was acting, and we tried all these usual suspect areas, no effect whatsoever. Brandon Hall did a lot, most of these local infusion studies. He was quite frustrated with it, and I said, try the hypothalamus. And he did, and he supramammillary nucleus, lateral hypothalamus. He did see some effects of reducing nicotine self-administration, particularly in the supramammillary nucleus of lacasterin. So that was the area of effect that we did see. Histamine, it's not just in your sinuses, it's not just in your leukocytes. They are in your brain as well. H1 receptors, in particular, we found to be important in terms of nicotine self-administration, significantly reducing the nicotine self-administration and acutely and chronically. And then in contrast, beta histine, which is an agonist of these receptors, significantly increase the nicotine self-administration. Glutamate with NMDA system. We did try ketamine. Amir Razvani was a leader in this study. It significantly reduced it. Nicotine self-administration. Ketamine is possibly a good therapeutic drug. It's not. It also has abuse liability, also has psychotomimetic effects. So maybe there would be another one that we could try. And so we tried memantine, which is a drug used in Alzheimer's patients. And acutely, interestingly enough, it actually increased nicotine self-administration. But we went on and did the chronic study in any case, and we found that it actually decreased it chronically. Not only during the period of its self-administration, during week 2 here you can see, but also after the end of the treatment, which is really kind of the stiffest test. Does your therapeutic treatment have residual effects after you stop the treatment? And this actually passed that test. Interestingly enough, if you break down those data day by day, you see the first day that we gave the drug, it significantly increased nicotine self-administration, like we found in our acute study, and then it decreased it after that. Norepinephrine, we tried idazoxin, adrenergic alpha-2 antagonist, and we did see acutely that it increased nicotine self-administration. So here we saw the opposite sort of effect. We don't have the data for chronic. Monoamine reuptake inhibition, bupropion, you've seen this graph before, but bupropion has an effect of decreasing as well. And amitriptyline, which is a triple reuptake inhibitor, which inhibits the dopamine, the serotonin, and norepinephrine reuptake, has an effect acutely as well as chronically. We've tried combined treatments together with nicotine. The D1 antagonists had some mutually beneficial effects in lacasterin, and nicotine likewise having mutually beneficial effects, and also likewise prolamine and nicotine having some combined effects. And amitriptyline potentiating the effects of nicotine, and nicotine potentiating the longevity of the effects of amitriptyline. And back to this graph I keep showing, varenicline and bupropion potentiate each other's effects. So I'm running short on time. So as with the summary, so as a validator of the model, the rat model, nicotine, varenicline, and bupropion all significantly reduce nicotine self-administration. And then extending from that, we found a number of new treatments that reduce it. Lacasterin, we saw in the rat model, it actually did predict the human effects. Amitriptyline, we found in the rat model, those human studies will be going on soon. We've also seen dextromethorphan, which I didn't show you the data. Those studies are ongoing at this point. the chronic nicotine enhancing the effects of lacasera and amphiptyphidine, and then local infusions showing not just your commonly accepted areas like the nucleus accumbens being important, but other areas like the hypothalamus and the posterior parietal cortex, insular cortex being important as well. Next steps, we're going to relate different neural systems and see how they correspond to these different behavioral systems in terms of appetitive behavior, habitual behavior, reinforced behavior, secondary reinforced behavior, stress alleviation, and how those relate to these different neurochemical and also neural anatomic areas and how they interact, and then determine simultaneous, which we've looked at, but also potentially sequential interactions where you may have a certain treatment before cessation, another treatment after cessation during withdrawal periods that may be most efficacious. And then looking at the how these can help people quit. Funding supported by the P50 from the folks at NIDA, and collaborators, Jed Rose, long-term collaborators, so Amir Razvani, who's here, has been key in all these studies, and some really very talented and devoted postdocs and technicians and undergraduate students and graduate students. And then I often show this map. It's not the ventral tegmental area up in some small lake in Minneapolis, near Minneapolis, and the ventral tegmental area in the fun town of New Orleans, but coming together of a number of tributaries and a number of basins. But really, it's not. This doesn't really. This analogy does not encompass it because it's not a final common pathway. It all ends in the lower Mississippi and down at the delta into the. I think it's called the hypoxic zone from this map. But there is this feedback, and potentially in terms of the feedback, the habenula, which has been mentioned before in terms of telencephalic feedback to the brainstem, I think is quite important. So these interacting systems are not beyond our understanding, and I think we need to kind of embrace the complexity to try to better understand, not just going down in more and more reductionistic of what particular molecular events are, starting the series of dominoes going down, but how be more integrative and how these systems interact to explain not addiction in the singular, but addictions in the multiple sense, and that we can devise treatments that are going to help people with not only tobacco addiction, but other sorts of addiction, opiate addiction, cocaine addiction, alcohol addiction, and so forth. Okay, thank you.
Thank you, Ed. We have some time for questions. First, I have to say that your data have shown so many individual treatments, potential treatments, and combinations in the animal work that look promising that I think we'll need a 20-year center grant to test them all in the human clinical trials, but there's a tremendous amount of promising material. Quick question, and then I'll turn it over.
From your mouth to Nora's ears. From your mouth to Nora's ears.
Just a quick question, while fresh in mind, on the automaticity, coming back to that.
Oh, yes.
What would you see as a good measure in rats of automaticity as apart from other motivated sources of the tributaries of addiction, like, for example, failure to extinguish, perhaps?
That would be one, although, I know enough in terms of behavioral assessment of animals not to ascribe voluntary versus involuntary actions to my animal subjects. They can't really report on that level. I would actually very much like to do some dorsostriatal local infusion work to get at some of the mechanisms possible in terms of these habits. Yeah.
So I have a question about what you think might be the mechanism of this time course of the effect of memantine with the acute augmentation of self-administration and then later chronically with respect to its action on glutamate, and how that might be related to what people are thinking about N-acetylcysteine, which is a metabotropic glutamate receptor agonist that ultimately results in reduced intrasynaptic glutamate.
Right. So, if you just withdraw the reinforcer, often there is an extinction burst where you would see an animal that's working, even if it's just working for sucrose or food, or for some stimulant, that if you withdraw the unconditioned reinforcer and you see an extinction burst of like, where the heck is it? It may be some of that in terms of trying to get back to the level of satisfaction that they once achieved. The other thing is it brings in this cognitive aspect, because memantine is known to have cognitive enhancing effects. So how it may be kind of starting to decondition the secondary from the primary reinforcer over time. But yeah, that's an interesting effect. Yeah.
Thank you so much. A very nice overview. So a question, I was wondering, for some other psychopathologies, like for example depression, there is this idea of personalized approaches to treatment, reflecting the fact that antidepressants are not effective in all patients in all the same way. So if you can identify people that suffer from particular type of depressions, then perhaps they will be responding to particular type of treatments. Then would you think that this kind of approach will be valuable for smoking cessation as well, breaking down smokers into subgroups of particular ways of smoking and motivations, and then specific treatment for those people?
I would think so, and it would be interesting to try to classify smokers into what they're driven by. Are they driven by the cues of smoking, or are they driven by the nicotine effects itself? Are they driven by a kind of more or less hunger-based desire, or are they smoking to alleviate stress, and that you may be able to attach that to it. The other thing we've been trying to do is to understand whether early response to a particular drug treatment might predict later efficacy on the individual basis, so that you might be able to not only behaviorally characterize someone, or genetically, I mean that's not my area, but other people do that, but also pharmacologically probe people who want to quit, and to see would you benefit from a serotonergic intervention, or intervention, or a dopaminergic intervention, or a nicotinic intervention, and then go with your best bet first. Yeah.
One more question now, and then we will have more discussion later, but now.
Just a note of information before I ask my question.
Yeah.
Before I worked in the brain and behavior, I spent several years in the liver.
Oh, I'm sorry.
And there's more than one cell type in the liver.
Yes, I apologize for...
Actually, there are 4. But at any rate, with respect to the question, this has puzzled me for some time, Ed, and I've never asked the question either privately or publicly, but I think this is a good time to ask it. You presented some data this morning that reuptake inhibitors for the dopaminergic system result in a decrease in nicotine intake.
Yes.
You've also shown that dopamine receptor blockers result in a decrease in nicotine intake. So one system is shutting the system down, the other one is increasing the activity of the system, yet the behavioral outcome is the same.
Well, none of these drugs are clean, and sometimes there's a virtue in their dirtiness and their multi-receptor action, their mechanism action. Like amitriptyline is a triple reuptake inhibitor, and maybe the effects on serotonin and or norepinephrine may predominate. The bupropion also has effects actually blocking nicotinic receptors, may be key in its effect. And the other thing is, back to the days when Jed and I were doing this agonist-antagonist approach, in any case, either blocking the receptor per se or blocking the reuptake, you're interfering with the fidelity of the system and the communication aspects of it, so that by interrupting that piece of the circuit, you may be achieving the same thing, and either turning down the volume or blasting the volume so much you can't hear the message. Do it again.
Your data that have been accumulated over a number of years indicate that, at least for reinforcement, the neurobiology is incredibly complex. Yet, at the beginning of your talk, you're talking about addiction being more than reinforcement and more than the alteration of dopaminergic turn in the nucleus accumbens. If you were going to try to develop an addiction to tobacco broad spectrum antibiotic, and I'm going to hold your feet to the fire, what systems would you attack first? Not system, systems.
Well, nicotinic systems are important, and dopaminergic systems are important. I think glutamate systems are really quite important. Even though I didn't present any data on today, next on the list, the GABA systems are quite important too. So I guess I would...
You're waffling there, Ed.
Well, you asked me for systems, and I gave you a laundry list.
Well, and in fact, harking back to, was it our 12th, 11th, 13th annual Duke conference, the title was GABA and glutamate, the yin and yang of nicotine reinforcement.
Yep. Okay, well, thank you very much.
Thanks very much, Ed. I will introduce our next speaker.